Okay, hello. So we'll start the fourth topic of immunology, which is the mediators of inflammation and immunity. So basically, we'll talk about two mediators of inflammation. The first one is the chemokines. So chemokines actually it's a very small protein, small peptides around the maximum 16 kilodalton, and they only released by injury, and they are very active at very low concentration. And all the chemokines actually share a sequence homology of around 30 to 50%. They can transmit the signals through a protein on the surface of the cells. And this protein actually, if you can see from the figure, it consists of 7 transmembrane protein. That's mean the structure of this protein in the cell membrane can transverse the membrane 7 times. And these kind of receptor activate and attract leukocytes once stimulated, once the chemokine stimulates cells having this receptor on its surface like leukocytes, it will be attracted to the site where the tissue damage occur. So chemokines actually can be classified into two subcategories based actually on 2 pairs of cysteine C. So either CXC chemokines, which is the alpha subcategory, and these subcategories used to attract neutrophils, and they are the most potent of these subcategory of chemokines are interleukin 8, IFN gamma, interferon gamma, and the platelet factor 4, inducible protein 10, and the MAF or macrophage activation factors. While the second subcategory is called CC chemokines, beta, the beta is the second subcategory, and they actually attract monocytes and T lymphocytes. If you attract as well eosinophils and basophils and NK cells, natural killer cells, via the macrophage chemotactic protein, the macrophage inflammatory protein, and regulated on activation of normal T cells expressed and secreted, which are normally abbreviated as RANS. So what about cytokines? So cytokines, it's a major, another big class of which chemokines can be a subcategory of cytokines. So cytokines actually is intracellular signaling molecules or proteins act locally in a paracrine or autocrine manner by binding to a very high affinity receptors. They have an overlap function. That's mean there are some redundancy in the function between the different cytokines. So you can see as a single activity can be caused by multiple cytokines, and vice versa. Multiple activity can be caused by a single cytokine. And this process, of course, is called beliotropism. Lymphokines, for example, are cytokines produced by lymphocytes, while monokines are cytokines produced by the monocytes or the macrophage. While interleukin 1, 6, and TNF alpha are collectively called endogenous pathogens, and they induce the MCP and interleukin 8 and the acute phase response. For TGF beta, they are actually potent wound healing and immunosuppressive agents, and they inhibit one of the very important cytokines, interleukin 2, which actually promote one of its functions. The interleukin 2 is promoting the proliferation of many cell types, and as well, one it's a very crucial function of interleukin 2 is to promote the switching of B cells to start synthesis of this type of antibody, which is called IgA. Cytokines receptors exist actually on many cell types. and they can circulate in the circulatory system. Actually, not the whole receptor circulate in the circulatory system. Only the extra cytoplasmic portion of the receptor, they can circulate in the circulation, and they can combine and block with the cytokine which is in the serum before it's reaching its cellular target. So it's a mechanism of inhibiting the action of cytokines in case the body is not wanting to have its action and it has been concluded. So the exoplasmic phase of the cytokine receptor can detach and circulate and bind to the cytokine, so it will inhibit its action. So this table shows some of the cytokines, or actually most of the cytokines and their action. So you can see two of the major cytokines is interleukin-1 alpha and beta, and interleukin-2. For interleukin-1, you can see that the major cell source of these interleukin-1 are macrophage endothelial cells, the one that lines the blood vessels, the dendritic cells, the one which do the antigen-presenting cells, and as well Langerhans cells, which is considered one of the type of the dendritic cells that exist in the skin. While the interleukin-2 are majorly secreted by T helper 1 cells, and we will understand later in coming lectures how the T helper 1 cells is very crucial and very important cell in actually activating the whole adaptive immune response, the T helper 1. So for interleukin-1, the major immunologic action actually stimulating interleukin-2 receptor, one of the major cytokine, one of the major receptors that respond to interleukin-2 that is responsible for proliferation, and as well enhance B cell activation, induce for interleukin-1 alpha and beta, induces fever, acute phase reactants, and the interleukin-6. It increases as well non-specific resistance, and they can be inhibited by endogenous interleukin-1 receptor antagonist. And actually, if you remember, this is actually the same we have just said, that the exoplasmic part of the receptor in the serum will bind with the cytokine to stop its action. While for interleukin-2, which is can be released from T helper 1 cells, actually it functions in T cell growth factor. That's why T helper 1 cell with interleukin-2, one of the major and the crucial cytokine released from very important cell to amplify and enhance the T cell growth factors. As well, it activates the NK and the B cells. So majorly, it's like the switch of the adaptive immune response. For interleukin-6, which actually secreted from monocytes, T cells as well, and the endothelial cells, they induce acute phase reactants as well, fever, and late B cell differentiation. And we'll understand later on what's late, what's early B cell differentiation. Then we have interleukin-7, which is exclusively secreted from the bone marrow cells, and they stimulate the and we have to say pre-B cells and pre-T cells. While interleukin-8, which is secreted from the monocyte, endothelial cells, lymphocytes, and fibroblasts, actually they are chemotactic factors for neutrophils and T cells. What about three other important cytokines, which are the of course, the other cytokines, each of them have a different function and related function. The other three cytokines are TNF alpha, TNF beta, and TGF beta. So for TNF alpha, actually it is secreted from the macrophage, T and B cells, and as well large granular lymphocytes. And this cytokine is very crucial, and because it's very cytotoxic for tumors, and it causes cachexia, which actually syndrome can be. It looks like the syndromes when someone is affected by cancer, like loss of weight, loss of hair, collectively. And as well, TNF-alpha can mediate a bacterial shock. While TNF-beta, which is secreted from the T cells, it's cytotoxic for tumors. For TG transforming growth factor beta, they almost secreted from almost all normal cell types, and it's an internal machinery that inhibit the proliferation. These cytokines will activate internal machinery to inhibit the proliferation of both T and B cells. It's a kind like a break, break for the immune system. It reduces cytokine receptors. It is a potent chemotactic agent for leukocytes, and as well it mediates inflammation and the tissue repair. So in this figure, you can see a very major difference between the chemokines and cytokines. Actually, chemokines controls chemotaxis in a directional movement, while cytokines control chemokinases, which is totally different mechanism. And actually, as well, it is a movement, but it is in random direction, okay? And this is one of the major difference between the action of a chemokine and cytokine. So here it's a simple, very simplified difference between chemokines and cytokines. So chemokines actually are superfamily of cytokines, as we just said in the beginning of the lecture, and they mediate chemotaxis, while cytokines are immune modulating agents and made up of from proteins. Okay, actually both of them are made of proteins. The first one, the chemokines involve in directing the cells in the immune system to the site of infection, while the cytokines generally involve in both cellular and antibody-mediated immunity in the body. The examples of the chemokines include CC-kines, CXC chemokines, C chemokines, and CX3C chemokines, and all structure types of chemokines, while the cytokines are very large family of molecules, including as well chemokines, interleukin, interferons, CSFs, TNFs, and TGFs are all types of cytokines in the body.